So for this section of chapter 15, we're looking at the third mechanism of protein sorting, which is transport by vesicles. So we're going to look at the process of using transport via vesicles, and there are two different pathways, an outward and an inward pathway. The outward pathway in this diagram is shown via pink arrows, and the inward pathway via these green arrows. So the inward pathway, this process is called endocytosis, and the outward pathway, it's called exocytosis. And there's a couple nice videos that are linked in our unit materials, one on basic endo and exocytosis, and another one about vesicular traffic through the cell that do a good job of explaining some of this. I just want to mention here that the outward pathway, right, begins with the endoplasmic reticulum and for proteins, and then they are shuttled via vesicles to the Golgi apparatus, and then two routes. They could either end up going through vesicles to the plasma membrane and be secreted from the cell, or they can end up fusing with what we call early endosomes, and then becoming the lysosome. So the enzymes that end up in the lysosomes would go through this pathway via an early endosome. And then there's also, again, the inward pathway of endocytosis, and vesicles from the cell membrane here end up with those in those endosomes again, and then those fuse with the lysosome, and the products are digested and the molecules distributed accordingly. Now, on the surface, that all looks great, very easy to follow, right? You've got an inward, outward pathways, a couple different options, everything's traveling through vesicles, but it's much more complicated, and we're just beginning to learn how it works, because vesicles have to take only what they need to transport, and they have to fuse with the correct target organelle or membrane. So there's many different types of vesicles that are carrying very distinct molecules, and these aren't only the molecules on the inside of the vesicle in the lumen, soluble molecules, but they're also carrying membrane bound molecules like membrane receptors and things that are held across membrane. So they're carrying that in their membrane, and they're carrying soluble proteins in their lumen. And so one thing that we've learned about these vesicles is there are what we call coated vesicles. And the coating seems to be different types of molecule. There's one molecule called clathrin we're going to look at that forms kind of a basket around vesicles and helps them actually form. So here in this set of pictures, you can see a budding vesicle. Let's see if I can get my pen. So here's a vesicle beginning to bud, and around if I can get my pen to show up, around the edge of the budding vesicle, let's see, here we go, around this edge you can see some molecules, kind of a line of thick line of some type of molecule, and these are those clathrin molecules. They form this coat around the vesicle, and it eventually pinches off, and we'll see how it does that, from the membrane that it's originating from, and becomes a separate vesicle with this coat around it. And then the coat will come off and the vesicle will travel to its destination. So the coat appears to help the vesicle kind of bud and pinch off from the membrane that it's leaving. So here's a cartoon version of those pictures, and this is showing the clathrin molecule in green. So it's a they call it triskelion molecule. There's some really great pictures of it in a video that I've linked into the unit materials, so I would look at that because it explains what's going on well. But clathrin is involved in coating the vesicles that both bud from the Golgi and take materials to the plasma membrane, and also vesicles that bud from the plasma membrane and take things to the endosomes. So clathrin molecules assemble in kind of a basket-like network around the vesicle, and it helps the vesicle bud from the source membrane, and it also helps the vesicle bring the correct transport molecules. So if you look here, clathrin molecule, look over here on the left. Let's see if I can do this. Can't see it again. Clathrin molecule here in green, there we go, is attached to an adaptin molecule, and that adaptin molecule is attached to this blue cargo receptor, and this cargo receptor grabs onto specific molecules that are to be transferred, in this case from the extracellular space into the cell. So this clathrin coat continues to form the vesicle and get larger and larger as it encircles the vesicle. And then there's this protein molecule dynamin that is a GTP binding protein, and it's itself a small GTPase, and it uses the energy of GTP hydrolysis to pinch off the last bit of membrane to free the vesicle. And then once the vesicle is free and in the cytoplasm, that clathrin coat comes off and the vesicle, sometimes called the naked vesicle, is transported. And that's usually via something we'll look at in a later chapter, the cytoskeleton, microtubules in the cytoskeleton. This table from your textbook shows you some of the types of vesicles, again, that clathrin is involved in forming. So over on the left, type of coated vesicle, clathrin, and again, this coating will help vesicles move from the Golgi apparatus to lysosomes via the endosomes, and also from the plasma membrane to the endosomes. And then there are other coated proteins. COP just stands for coat protein, but it's a different one from clathrin. And so there's a whole family of these things, whole group, and they're involved in, transporting things between the ER and the Golgi, and between different Golgi compartments, and between the Golgi and the ER. So this is the other end of that process. How are vesicles targeted to specific organelles? And this is kind of a two-step process, as what we understand now, and it involves, again, GTP binding proteins, or GTPases, these enzymes that will hydrolyze GTP and release energy. But this is showing a vesicle that's coming from the ER, up here on the left, and that vesicle is going to fuse, say, with the Golgi. And at this point up here, let's see, it's a naked vesicle, so it doesn't have the coat protein on it anymore. It's carrying its cargo in brown here on the inside. And on the outside of this vesicle is a protein called RAB. And this is the GTPase. So, I don't know if I can write with my mouse very well, but GTP is bound to this protein. So the first thing that happens is there's a tethering protein from the target membrane, this green long protein that will attach to the RAB, the GTPase, and that starts kind of a timer in a sense. The GTPase is going to work to hydrolyze the GTP, and when that happens, the RAB will disconnect from the tether. So before that happens, these other molecules called snares, there's a target snare that's called T snare down here, and then there's the vesicle snare that's called the V snare. And once the tether brings the vesicle close into the target membrane, these snares will contact one another and begin twisting together. And that's going to provide the force that's going to allow the vesicles to fuse with the target membrane. And so if this isn't if the snares don't match very well, like if a vesicle's going to the wrong organelle, then they won't be able to complete this interaction before the GTP on the RAB, the GTPase of the RAB can hydrolyze, or sorry, hydrolyze the GTP to GDP. So that's why that kind of acts like a timer. If the pairing is good, the snares will pair and wrap together before the connection of the RAB is lost with the tether. If the pairing's not good, then they won't be able to do that, and the vesicle will be released. So this slide is showing how the snares, there's more than one per vesicle, can facilitate the fusion of the membranes together, because that's not a very energetically favorable process, because water has to be displaced from all the hydrophilic heads of the phospholipids. So the V snares and the T snares wrap around each other, getting the kind of forcing the water out and getting these membranes to come close enough together so that they can fuse. And there's a video of this process in your unit materials that you should watch as well.